So, you know that VLDL are those lipoproteins which are relatively smaller than the chylomicrons, because they are relatively denser as compared to chylomicron, but still they have a really very large amount of lipids as compared to the other lipoproteins. It is produced in the lipid, is composed predominantly of triacylglyceride, which makes 60% composition of these of the composition of the VLDLs. As far as the function of VLDL is concerned, of course, because it's moving the lipids from one part of the body to the other, so it will carry lipids from liver to the peripheral tissues, and the lipids which are then moved through the peripheral tissues, they are acted upon by the lipases, the LPLs, lipoprotein lipases, as a result of which the specifically the TAGs, the triacylglycerides, they are degraded. As far as the secretion of the VLDLs by the liver is concerned, keep in mind that it is secreted into the blood after coming from the liver in a nascent form, as we saw the chylomicron also during their very starting phase of formation, that was also a nascent molecule. Similarly, the VLDLs in their very early stage are in their nascent form, and the apolipoprotein they contain is the ApoB100. In contrast to the ApoB48, here it is ApoB100. Of course, you can understand why it is ApoB100, because the ApoB protein, which is encoded by the gene ApoB, it's 100% of the same gene, not the 48%. During its passage in the blood, it's moving from one part of the body to the other, they obtain the two other apoproteins, apolipoproteins like ApoC and ApoE from HDL. So this is what I have already told you. This transfer of proteins or apolipoproteins from one category of the lipoproteins to the other is a very normal thing. It's not something which occurs because of any abnormality. The ApoC is required here also in VLDLs also. It's required to activate the LPLs, the lipases, which will then degrade the TAGs. As a result of this, as we saw in the chylomicron, more the TAGs gets decreased because of the action of the lipases. The compound will the lipoprotein will become more denser. So LDL will become more denser because of the VLDL will become more denser because of the action of lipases, because it's degrading the lipids. Then once they have activated and performed their function, the ApoE and ApoC are returned to the HDL, the high density lipoprotein, while the VLDL retains the ApoB100. As I have already told you, the exchange is a normal thing. It goes on between HDL and VLDL, or HDL and LDL, and so on and so forth. Because the density is increasing while it's moving from one part to the other, the VLDL, what will happen? It will be losing more and more TAGs and becoming more and more denser, and decreasing in size, and ultimately it will be changed into LDL. Some of the abnormalities also occur. I mean, some of the abnormalities in the formation or transportation of VLDLs leads to some real problems, real medical problems, like familial type 2 hyperlipoproteinemia. And also, of course, you can understand why this hyperlipoproteinemia will occur, because more and more accumulation of the what we say, the more and more accumulation of the triacylglyceride, because the movement is not taking place as it should have been taking place, and also the susceptibility to the Alzheimer's disease. And you can think how it could be, how Alzheimer's disease is a brain disease, is a neurodegenerative disease, which involve many genetic factors. But how this lipid transport mechanism can be, or defect in the lipid transport can be linked with the Alzheimer's disease? Yes, you can think about it. Yes, you are right, because uh, the accumulation of lipids can lead to cell death. And if neurons are dying because of accumulation of excessive lipids, because of the defects in the transportation of lipids, it will lead to the death of the neurons, which one of the consequence of which could be the Alzheimer's disease.